for you and the other organizers for inviting me. It's my first time down to Australia, so happy to be here. I also want to start by acknowledging my colleague, Malia Guillen. All the stuff I'll talk about is done in a tight collaboration with her and her group. She probably would have been here herself, but she just had her first child about a week and a half ago, so she's doing well. So I run the Bioinformatics group at the Danforth Center, and our goals, in addition to sort of providing typical bioinformatics services to people at the Danforth Center, are to build enabling technologies for plant science. So most of that work is in this area of plant phenotyping, but we also do some stuff with genomics and other things. My approach is I like software that's community-driven. So it's not just me sitting in a room by myself writing software. I like to have like community input, so that all the stakeholders are involved in the process. And then I also have the ethos that we want these tools to be reusable, repurposable, and extendable for ourselves and for everyone else. And then ideally, too, these are tools that can be used in training and outreach, because I think, especially in plant phenotyping, there's a real strong visual, tactile connection with students that is valuable. So I'll talk about two things today. First, some of the things we've done in this area of low-cost solutions to plant phenotyping, primarily Raspberry Pi, platforms for imaging. That's work that's kind of led by this ad hoc maker group we have at the Danforth Center. So that's people that are interested in that do-it-yourself kind of movement of prototyping, building your own sensors and things like that. And then I'll talk about some of the software tools we have to analyze data from our expensive systems and our low-cost systems. So obviously I won't talk about these too much today, but we have many not so low-cost solutions at the Danforth Center. Started in 2013 with the Bellwether phenotyping facility that we built at that time. It's a Lemnitech Scanalyzer system. We have some specialty systems now, like X-ray CT and Malia's hyperspectral imaging robot. That was the Lemnitech system. And then all the way to our field system in Maricopa, Arizona. So the Bellwether platform and the TerraRef platform are both, they can screen large numbers of plants. They have multiple sensors integrated on them, and then they obviously have a high level of automation. So that, the high cost comes with all those benefits of being able to turn on the system and run it without having to think about the engineering. But obviously fairly high cost of running experiments on them. Pretty time consuming to set them up. And the TerraRef system, for instance, is dedicated to essentially one project, so it's largely just growing sorghum year-round. And then the Bellwether system, because it's quite popular to use internally, if you want to do an experiment on it, you have to think a long time in advance about getting access to it, setting up your experiment. So one of the things we realized early on is it'd be nice to be able to image all of your experiments all the time, right? Like, what if all the growth chambers in our facility had cameras in them? So luckily, around the same time that we were building the expensive stuff, the Raspberry Pi computers became quite popular. So these were introduced by David, so I won't go into it too much, but they're $35 US computers, so they're a complete system on a chip. The only additional things you need is a power cord and an SD card to store the operating system on them. So they run full Linux operating systems, so they're essentially autonomous from that point of view. And then you can attach all sorts of accessory components onto them. You can attach all sorts of sensors that you can pre-buy, or you can build your own sensors. And then importantly for us, there's this 20 or 25 US dollar camera module that you can plug in. And so for about $100 US, you can have a fully Linux controllable camera. So that lets you do automated data collection at pretty low cost. And then the way we use it primarily is to image whole like trays of plants at once. So you're capturing data on, say, 17 or 18 plants all at one time. Another nice thing about them is you can buy the camera module in a version that doesn't have an infrared filter on the camera. And so you can, the camera is then sensitive to infrared light. So in this case, this is a movie from Dmitry Nusinow's lab. They're interested in diurnal patterns and circadian patterns. So in this case, they have put a visible light filter block in front of the camera, so the camera can only see infrared. And then they have a custom soldered LED panel behind this auger plate with a diffuser, so they can watch seedlings grow for 24 hours a day. In the background, there's like a normal diurnal light cycle going on, but since the camera can only see infrared light, you just get this consistent infrared illumination all day. So when you have a commercial system where you have like a plant to camera or a camera to plant type setup, you have that key component, which is the automation built in. So you can have more expensive sensors because you can either move the plants to the sensors or move the sensors around. You could do that yourself too, but in our case, what we do is we figure that the Raspberry Pi camera systems are low enough cost. We can just have one camera for every unit we want to image, which in our case is a tray. But there's a cost to that from a complexity point of view, which is if you have hundreds of these or more, you have to start worrying about how to control all of them at once. So some of the things we do to take care of that is you can clone the SD cards. So once you configure one, you can just clone that operating system and plug those clones into every machine. Or you could do fancier things like boot the Raspberry Pis over a network from a master image. Or some of the stuff I'll talk about is remote configuration. But there's other complexities like actually just managing power. You're talking about like power cables, potentially network cables going to every Pi. It gets a little bit big and heavy at some points. And then data management also starts to become a bit of a problem when you're collecting data from hundreds of cameras all at once. So what we do is when we set up one of these systems, we have our IT department record all the network adapter addresses into our building network systems so that we can get static IP addresses for all of these things. That way we can know, based on their addresses, like where we're putting them. So once that's set up, that's a bit tedious at the front end. It's not so bad after. Okay, so one of our setups, a fairly early one, which was interesting, was 180 of these Raspberry Pi cameras on the ceiling of a greenhouse. And they're all Wi-Fi controlled, so the only cable going to them is power. And that turned out to be actually quite a challenge. So the Raspberry Pis don't have particularly strong Wi-Fi networks, or these are external antennas that are plugged into these. So there's a ton of Wi-Fi interference with this many systems. There's 4 Wi-Fi access points powering all these things. And they also have a very complicated schedule where they actually turn off their own Wi-Fi networks based on when they're imaging. Otherwise, the interference is just so much that it's quite difficult to control them. So this group has them set up in stereo camera pairs, and so they can do full 3D reconstruction of the greenhouse. They're doing that once an hour, which probably is excessive, but but it's doable with this kind of automation. So the way one of the innovations that they had come up with to control all these and configure them is to use a system called Ansible, which is an IT automation product that's freely available. It basically lets you control Linux machines, or maybe others, through secure shell, but it can let you submit like commands to these remote machines so that you can make them all do something at once, but you can also do other things like make sure all of the machines have the same configuration files. So if you want to change something globally, you don't have to log into every single machine. You can just send one command from a central point and reconfigure them all. So sort of the second generation system that Malia's lab built, which definitely learned some stuff from the greenhouse installation, was this dual growth chamber setup where there's 72 cameras in each growth chamber. So there's one camera per tray, and then the growth chambers are set up where one's normal control temperatures and the other one can go to low temperatures, I think 4 degrees Celsius. So she can kind of do these temperature stress experiments. So she's using the same Ansible software that the other group's using, but what she did, since her Raspberry Pis are a newer generation, they work with a setup called Power over Ethernet. So we have a switch outside the growth chambers, which you can see all this massive cables here. But the Ethernet cable is transmitting both data and power. So there's still just one cable for every Pi, but no Wi-Fi, it's just Ethernet. So the data transfer happens in like 15 minutes once per day, even though they're taking an image every hour. So that it's a little more expensive to buy that kind of switch, but it was definitely worth like the effort in not having a crazy Wi-Fi network. So if we actually look at the comparison to, like, say, our Lemna Tech facility versus Malia's, she can grow about twice as many plants as we can in the Lemna Tech system. I mean, granted, they're small plants. And she can actually take quite a bit more images per day. So in the Lemna Tech system, we're doing 3 RGB images per plant once per day. So that's 3,400 plant images, 3,400 plant images per day. Versus hers is about 2,000 a day, so it's on the same scale of data collection. But if you think about per plant, it's imaging the plants 36,000 times per day. So in a lot of ways, it's actually a comparable amount of data, but more information on more plants. Although, we don't have automated watering and stuff like you'd get with the other kind of system, but the cost is obviously much lower. I think her whole setup was around $20,000 US. So for the next part, I'll talk about like how do we analyze this data. Whether it's from our commercial systems or from these low-cost systems. So we developed our own image analysis package called PlantCV. It's actually a wrapper collection of a lot of other tools for software development and Python-based image analysis packages. So it's a modular library of tools that you can plug together the modules to form your own workflows to analyze your images, and then parallelize workflows across your data set. So the way this looks would obviously briefly take like an RGB image, say convert it to some type of grayscale color space that would ideally be selective for plant pixels, apply a threshold, apply many other steps to filter out background pixels, and then once you have your plant object, as we all know, you can then measure the physical properties of the plant, or shape, color, things like that. So then once you have one of those workflows, we have some tools to let you analyze your data set in parallel, given that workflow. The nice thing about this is it's flexible, so if different kinds of imaging setups or different kinds of cameras will have their own quirks, so you can kind of design your own workflow to work around your particular problems. Like I said, ideally, we do this through community development, so it's not just us. So Malia and I are the primary developers, but we've worked with a lot of other people on this project over the years. So these are the people that we know. There's also a fairly decent community of people on GitHub that work with us that we actually don't know. We know their GitHub usernames, and that's all we know about them. But we've worked with some people, largely at the Danforth Center, and then some collaborators like Arhelia Lawrence's group from Arkansas State, and others. So I'll just briefly go through some of the features that we have. We're cross-platform. Mostly we use it on Linux systems, but other people prefer other platforms. Another key feature for us was integrating PlantCV with Jupyter. So Jupyter notebooks are a live coding environment, like interactive coding environment. So in this video, I'm just showing how I'm opening a Python notebook in Jupyter with PlantCV code loaded, and I'm just running it all. The cool thing is you can run snippets of code one cell at a time, and you can see the outputs of that snippet. So in this case, PlantCV is designed to print out the intermediate images, so you can quickly prototype a workflow in PlantCV using Jupyter, and then you can export your notebook into a Python script that then fits into our parallelization tools. So that's as close to a graphical user interface as we have, but it works well for us for this rapid prototyping. Another key feature for us, and it's not so much about usability, but about stability, is some of the tools we've built into on the back end for software development. So that's so that you, as a user, you would know that you're working with a stable version of PlantCV. And then we can also integrate feedback from multiple people all at the same time. So the way this works is if you're a developer, whether it's me or anyone else, you start with your own local version copy of PlantCV, and you make a change. You submit it to us as what GitHub calls a pull request, which itemizes like what you've changed versus the existing code. And that triggers a bunch of automated processes. So the first is a automated build test in a service called Travis CI. So that runs about 100 or so automated tests that we have. So we have known inputs and outputs, and we just ask, does everything in the code still work the way we think it does? If it does, then it runs coverage analysis, which looks at how many lines of code were actually tested, because if you don't actually test any code, your build will pass, but won't do any good. So we're up to like 94% on that, which is pretty decent. If everything looks good at that point, we can merge that code into the main version of PlantCV. It rebuilds the documentation automatically, and then that becomes the latest version. And then we also will make periodic static release points as bookmarks, and then those are archived on Zenodo automatically. I won't go through all these. We have a lot of basic image analysis methods built in, so you can do all the kind of usual things like conversion between color spaces and thresholding and different types of measurements on color, plant height, shape, size, things like that. We have some calibration tools built in, so like size marker calibration if you want to calibrate your measurements to physical dimensions. In this case, we're looking at seed image analysis. More recently, we added a color calibration tool. So if you have, in this case, we've got the X-Rite color card in our pictures that we can use to calibrate the color of every image if we want, or we have it set up so you can just periodically take standards and build color correction matrices. So for us, this helped in our Lemnitech system. We actually discovered that the lighting in the imaging cabinet varies in quality quite a bit over time. or time of day. And so you can get this noise in the color profile that we can correct out using these color cards. I'll skip over that for now. Another one that we've worked a lot on is some machine learning tools. So the one we have in there now is a statistical classifier. One of the main things that came about using it was a group that was interested in identifying the pustules and chlorosis associated with wheat rust. So they had these nice scans of wheat leaves, of a population of wheat infected with rust. So they wanted to be able to identify the different components of the disease. So there's a zoom in. So we had already had the statistical classifier, and so what we did was we figured, well, we just need samples of the colors of the different arbitrary number of classes that someone wants to define. So we just had them use ImageJ to do pixel picking, collect the RGB values from the pustules, the chlorosis, the leaves, and then the background as 4 classes, build a table of those RGB values, and then we use a kernel density estimation analysis to build probability density functions for hue, saturation, and value color properties. And then we just use Bayes' theorem to calculate the joint probability that a pixel is plant or chlorosis or et cetera, assuming that they're independent. So then given the RGB value of any pixel in an image, we can predict which class it's in pretty well. It just takes about 30 minutes to collect the training data on a few images, as long as your imaging is pretty consistent. So I don't have the data here, but I've been told by them that it correlates really well with their qualitative scale that they use by eye. But they can do additional things now, like measure the size of the pustules and things like that they couldn't do before. We've also used this tool to segment anthers from maize tassels for another group who's interested in looking at maize fertility mutants. Right, so when we built PlantCV originally, we had designed it to work on this 1 plant per image setup from our LumnaTech system. So obviously with Raspberry Pi imaging the way we're doing it, we get multiple plants in a picture, but we need to measure them separately. So we had to build in some tools to separate the individual plants first, then do our standard analysis second. So we've got that built in now, so we can measure the plants independently. We can keep track of the genotype information that the user provides, and then can measure those, the differences between genotypes as an example here. So the software is tracking that metadata like from the beginning where the user has the information all the way through to the end, so that you can correlate your measurements with that human supplied metadata. Come back to this. So I guess briefly I'll just say that we had some tools that a group had added for leaf segmentation based on a watershed segmentation approach, which works okay. It's a little bit tough to parameterize, and it can be kind of messy. So more recently, one of the things we've been trying to do is use deep learning approach for this instead. And so you can consider some different strategies for deep learning, like classification. Does this image have balloons or not? Semantic segmentation, which is which pixels are balloon pixels, which pixels are not balloon pixels. Or how many balloons are there? So you could do object detection. You don't actually know, you know where in the images the balloons are, and you know how many there are, but you don't know specifically which pixels are balloons. Or a sort of newer approach, which is a combined method called instance segmentation, where you can get both the location and the segmentation of that object. So because this community has developed some of these shared labeled resources, like the Leaf Segmentation Challenge dataset, that's actually a data set we can directly use in this kind of approach for instance segmentation. So we already have these labeled leaves, and we can feed them into one of these systems. So the one we're using is called mask RCNN, which is based off of a somewhat older technique called faster region-based convolutional neural networks. It just adds an additional step where once it finds regions, it can segment the object from that region. So we can feed in the original image and the labels, and then it will output masks for each object. And so this is an example where you can actually see that maybe it's a little hard to tell, but this image wasn't quite labeled correctly, so this leaf wasn't fully labeled in the label, but the algorithm actually sees the full leaf. So it's counted as a false positive here, but in reality, it's actually doing a better job than the human, in some sense. So we're still working on this. What we're doing is training these on Amazon so that we're not making that investment, the expensive investment of buying the GPUs locally. We then train a bunch of different models, and we build an ensemble model by averaging the weights from the different models. So we're getting a mean average precision around 0.6 at the moment. Right, so once you've run PlantCV, then you have a bunch of measurements in a database, and so then there's still, the next part, which is making biological inferences from that data. So at the moment, we have some tools for exporting the data to R. That's our preferred environment for analysis. But the data is easily imported into a lot of other tools that are available, like MVApp, HTTP mod, or any other system you really want, because you can just export the data as a text-based format. And I'll briefly say for parallelization, what we're doing now is doing this on a single machine with multiprocessing, which is nice because it's portable. Any system that has more than one CPU you can parallelize on, but it doesn't scale very well, so it depends on how big your computer is. So one of the things we're working on now is building more system-agnostic parallelization scheme. So we're doing that using a system called Nextflow, which is really cool. It basically lets you write an abstract analysis workflow that can then run on any queuing system or cloud system with or without containers, which are these virtual packaged environments. And so as an example here, I've got a link to an analysis workflow for Alex Bushk's DIRT image analysis software that we wrote in Nextflow. So the user can basically just say, run workflow star.jpg, and it will fully parallelize DIRT image analysis across an entire cluster. I'll go ahead and skip that part for now, and I'll just leave you with acknowledging my collaborators, many at the Danforth Center and elsewhere, and I'd be happy to take any questions.
Thank you very much for this